because I hit the laminin through an accident. Failures outweigh, the successes. And I remember they were jumping all over me on this. This is just artifact. I would prefer not to have to write grant applications. You go from a state of simple binding to avidity, where avidity is collective binding. One of them is looking at me right now. Just what's going wrong in the brain. If you're not good at troubleshooting, it's hard to make progress. I kind of wish there was a bigger basement membrane section in it. No, there is no best. Come on. No, my first love, oh yeah, okay, because I hit the laminin through an accident. My name is Peter Yurchenco. I live in New Jersey, and I am a professor at the Robert Wood Johnson Medical School, which is now part of Rutgers University. Science. Laminins, of course. It's an incredible molecule. It's huge. It's got 3 subunits. The larger versions, the larger variants, have 3 short arms and 1 long arm. It has multiple functions, and it integrates those functions. I think a lot about basement membrane assembly, which I work on, has to do with proteins that have multiple domains on them, each mediating a different function. And those functions have to work collectively in order to create a functional basement membrane. So in the case of laminins, or let's just take one of the laminins, laminin 111, it has to anchor onto receptors. It helps to have a little bit of extra glue, for example, through sulfatides, just to accumulate on the surface. But that anchorage then establishes a connection to the cytoskeleton, mainly the actin cytoskeleton, but not just. And it polymerizes. So it forms a planar polymer on the cell surface, which I think of as an initial basement membrane, a sort of immature basement membrane. But the other thing it does is that it binds to, you could say it recruits, other basement membrane components then to complete the assembly. And that assembly is completed substantially by the addition of type 4 collagen, which is the only other polymerizing component. And it is attached onto laminin, at least for stability, not necessarily to guarantee co-localization in the tissue, but it is attached through nidogen and possibly through heparan sulfate proteoglycans, although the evidence for the latter is not very good so far. It's a story also about cooperativity of assembly. You can have a series of weak associations collectively giving you basically strong bonds. And I think that the one thing that a polymer does is you go from a state of simple binding to avidity, where avidity is collective binding. And if you think about it in terms of thermodynamics, it is much harder to get dissociation if you're attached at multiple sites than if you're only attached at one. And the other thing about laminins is the remarkable things they do in their absence in development and in mature functions, basically. So early on, it's fairly early on, this was in the 90s, we became aware of these muscular dystrophies of a genetic origin, and muscular dystrophies that also have neuropathies and also have brain dysfunction associated with it. And when one of these, and there were mouse models for it, and one of them is called the DY2J mouse, turned out, this came from the work of Kevin Campbell's lab and also Eva Engvall. But it was the discovery that the DY2J mouse, which was a dystrophic mouse, it was a mutation in the LN domain, which caused an in-frame deletion. It's a splice donor defect. And as a result, then we found that the domain degrades further. It can't polymerize anymore. Wow, here we have a dystrophy only due to a laminin polymerization defect, right? And that later became a model then to use to try to go ahead and treat, to see if it might help that fraction of patients who have only mutations in the LN domain in which the rest of the protein is still expressed, assembled and expressed. So what that enabled me to do is to kind of move from just sort of very basic, simple biochemistry up through culture systems, organ cultures, which we did to look at the peripheral neuropathy, up to animal models, and then finally to start thinking about whether you might actually be able to do something about it. And that last part is still very much in progress. And then I'll come back to just one other thing, and that's type 4 collagen. All right. So that's how I got into the field. It was when I was a postdoc at Yale. And this was in the very early days. About the only thing we knew about basement membranes pretty much came, at least biochemically, came from the EHS, the Engelbrecht Holmes swarm tumor that was available at the time. And there were a series of purification steps you could go through to finally get milligram to even gram amounts of laminin 111, of nidogen, which was usually came out bound to the laminin, and type 4 collagen. This is the alpha-1, alpha-2 collagen. And that enabled self-assembly studies. That's how I hit on laminin polymerization. But the first thing I did was take this type 4, which is mostly dimeric as you extract it, and just warm it up in phosphate buffered saline. And lo and behold, it made these incredible networks that you could visualize by rotary shadowing. Yes, it had been shown just a couple years earlier. My advisor, Heinz Furthmeyer, was one of the people on the project, that there are these 7S N-terminal interactions in which 4 triple helices overlap by 30 nanometers and form a strong bond, it's covalent ultimately. And NC1, C-terminal domain interactions that give you a dimer, a trimeric dimer, as opposed to a trimeric monomer. But in addition to that, I was seeing all of these lateral associations. And story is, I was first challenged. I gave my first talk on this. It was, what was it, the 82 Gordon Conference in Basement Membranes, and Rupert Temple and Klaus Kuhn were at the meeting, and I remember they were jumping all over me on this. This is just artifact. I said, well, maybe. So I went back and I started collaborating with this guy at Dartmouth, George Rubin, and because he had this high-angle, thin platinum coat method of looking at structure, which we needed a real basement membrane to look at. And I picked the human amnion because you could slough off the cells and have it exposed as a big sheet. And so working on this, lo and behold, there too, we saw the lateral associations. Not only there was supercoiling that you could visualize in regions. And if you think about it, so here you've got, these collagen. For us, they were secreted, I think, mainly as dimers. We weren't seeing monomer to dimer conversion in vitro. But the dimers would, they would begin to form the 7S bonds. With this coiling, and I decorated it with anti-type 4 collagen gold, and could decorate the network. And at this point, there was another meeting, another Gordon conference, and I had all these stereo images. And I remember the first night, this was at Plymouth, New Hampshire, where in the summer it can get really hot and just filled with mosquitoes that are hungering for you. And Klaus Kuhn was there, and I said, come here, let me show you this. So I took him back, to my room. I had these stereo glasses, and I said, look, this is what I'm seeing. And at that point, he became, I think, my biggest promoter for the model. And so that is really how I entered the field, basically. And it's kind of been a gas ever since. Yes, there have been plenty of failures. I would say the failures outweigh, the successes in general. But the discoveries have been adventures. And I would prefer not to have to write grant applications, but there doesn't seem to be any choice in the matter. And so, here I am now, I guess pretty late in my career. And I'm focusing mainly on the dystrophy, and also trying to learn things like, I'd like to learn just what's going wrong in the brain, because they have seizures, all right, the mice. And the treatment we use, at least on the DY2J mice, stops, prevents the seizures. And what you see by immunostaining is there's a big drop in laminin alpha 2, even though it's only a polymerization defect in the brain, basically. So what's it actually doing to the brain? I'd like to know. Anyway, I think I still have a few more years left. I look forward to collaborating with people who know the systems better than I do. One of them is looking at me right now. And that's Sergei Budko, who I just think is a fabulous scientist. And let's see where things go. I never left. I think, first of all, I think you need both passion and patience. Things often don't work initially. You have to be ready to troubleshoot as you go along. I think if you're not good at troubleshooting, it's hard to make progress. And you have to be curious, I think curiosity may be, a really critical ingredient, basically. I think it was really good when the society formed. It formed really out of the connective tissue clubs that were located, like there was an East Coast one, there was one in the Chicago area, Midwestern one, there was one for California, and I don't know if there was one in the South or not. And I used to go to those meetings, and they would have fewer people assembling. So I think it was a good idea to coalesce it, to bring the group into a much bigger group, a group that would have, I think, more influence in the field. I don't think I have a particular meeting that comes to mind as being head and shoulders over the other, but. I think it's generally been well handled. I kind of wish there was a bigger basement membrane section in it. Ever since we lost the basement membrane Gordon conference years ago, because we didn't quite have enough people to satisfy the Gordon people. But aside from that, and I think that that's probably more on us than on the society. To increase the fraction of people in the field, and I think there's a bright future. So.